Dear students, today we will have another lecture, techniques of sterilization, that is important topic in plant tissue culture, and some microbiological experiments, how sterilization has to be done and why it is done, how microorganisms are restricted to make growth of particular tissue. So we have taken here few methods of sterilization which prevents microorganisms to be grown without our permission, because if microorganisms grows, then our tissues and our object which we want to culture will not be fulfilled. So there must be very much clear that we have to sterilize the system which we are using. There are four main methods which are employed to sterilize different materials needed for microbiological works. The techniques are sterilization by heat, sterilization by chemical treatment, sterilization by radiation, and sterilization by filtration. We'll take one by one that how sterilization can be done by all these four methods. The sterilization process include some physical agents like heat. Heat may be of two types, that may be moist heat or steam or dry heat. Techniques used, that is steam under pressure is autoclave, that is boiling, pasteurization, hot air oven, that is used for dry heating, flaming, incineration. These all are the heating methods which may be wet heat or it may be dry heat. Another method, that is by radiation, and radiation includes the non-ionizing and ionizing type. The non-ionizing radiations are the UV rays or infrared rays, whereas ionizing radiations, that are gamma rays and high energy electrons. The filtration technique includes the candle filter, asbestos spade, membrane dish, etc. The chemical agents, mainly the alcohols, aldehydes, dyes, halogens, metallic salts, phenols, and surface active agents, which may be used by spraying, dusting, or giving aqueous treatments. The gaseous agents includes ethylene oxide, formaldehyde, beta propanol lactone, that is used by fumigation. Now, how sterilization by heat is done? The heat is most widely used lethal agent for sterilization. Objects may be sterilized by dry heat applied in an oven and atmosphere of air by moist heat provided by the wet steam. Of the two methods, sterilization by dry heat requires much greater duration and intensity. Heat conduction is less rapid in air than in steam. Dry heat is used principally to sterilize glassware and other heat stable solid materials, but steam must be used for the heat sterilization of the aqueous solutions, some culture media, or some non-volatile agent. The autoclaving is the method by which we generally sterilize the culture media, the aqueous solutions, and it is the method of steam sterilization which is carried out in a metal vessel known as autoclave, which can be filled with steam at a pressure greater than atmospheric pressure, and sterilization this be achieved at temperatures considerably above the boiling point of water. The laboratory autoclaves are commonly operated at steam pressure of 15 pound per inch square above atmospheric pressure, which corresponds to the temperature of 121 degrees centigrade. The autoclave is a cylindrical metal vessel having double walls around all parts except the front to withstand the high steam pressure. The base of the inner cylinder is concave, which contains water. The vessel is heated by immersion heater fitted at the base of the autoclave. There is a strong lid covering the cylinder which can be tightly screwed. The lid is provided with pressure gauge, safety valve, the steam outlet and metal handles. Inside the vessel cavity there is a wire basket with triple stand where the materials are packed before the start of sterilization. Here we can see the autoclave, and we can see here that this is the safety valve, this is pressure gauge, this is exhaust valve, it is a screw bolt on the lid, and basically this is the lid. Then inner cylinder is there, that is having the inner body wall, then the outer jacket which is surrounding the autoclave, then it is steam flow and immersion water heater that is fitted at the base of the autoclave. Then here water is filled in it, and this is the foot of the autoclave. So these all are the different parts of autoclave which are used in microbiological and tissue culture experiments. The techniques of sterilization includes how the autoclave works. At the start of operation, all the air present in the chamber must be expelled and replaced by steam. This is achieved by the use of steam trap, which remains open as long as air passes through it, but closes when the atmospheric pressure consists of only steam. In some air remains the sterilization chamber, the partial pressure of the steam will be lower than indicated on the pressure gauge, and the temperature will be correspondingly lower. For this reason, an autoclave should always be equipped with both of temperature and pressure gauge. After adjustment of pressure, 15 lbs per inch square inside the autoclave, the pressure is maintained for 15 minutes and then autoclave is cooled. The another technique of heat sterilization, that is hot air oven. It is an electrically operated oven used for sterilization of glassware, with petri dishes, flasks, tubes, pipettes, etc. The apparatus consists of a large rectangular copper base and covered with asbestos sheets. It is also provided with a door and erected on a 4-legged stand. The roof is provided with a hole through which a thermometer is fitted inside for recording of temperature. The oven has 2 or 3 cells. The oven is heated by electrically operated heater fitted at the base of the instrument. There is regulator of heater to control the inside temperature. In figure we can see the different parts of oven. That is exhaust on the upper side of the oven. Here it is seen, and the diffusion wall is there, airflow damper, glass wool insulation is towards the body, and the turbo blower and motor is fitted at the base of the oven. Before sterilization, the glassware are dried properly and wrapped in brown paper and then exposed to hot air inside the oven. After loading of glasswares, the oven is switched on 160 degrees centigrade. The temperature will increase slowly up to a desired point where it will remain steady. Then at 160 degrees centigrade, the oven is kept for an hour. This is the appropriate temperature for sterilization of glasswares. Then gradually the temperature is brought down, and thereafter sterilization is complete. The red hot heat, the procedure is generally followed during working inside the culture room with inoculating needle. The inoculating needle is flamed to make red hot. At that high temperature of the flame, all the organisms present on the needle are killed. Sterilization by chemical treatments. Many of the substances used in preparing culture media are too heat liable to be sterilized by autoclaving. For such substances, a reliable method of chemical sterilization is extremely useful. The essential requirement for the chemical sterilization agent is that it would be volatile as well as toxic, so that it can be readily eliminated from the object sterilized after treatment. Alcohol, Dettol, Lysol, ethylene oxide, and here in tissue culture we can have mercury chloride and twin 20, twin 80, and the hypochlorite, sodium and potassium hypochlorite are also used to chemically sterilize the explants. So various chemicals are used for sterilization methods, and they are used in different amounts for different time, so that the explant is get sterilized after picking them from field. Sterilization by radiations, in recent years various kinds of ionizing and non-ionizing radiations also employed for sterilization of the culture room equipments. The exposure to UV radiation for a period of 45 minutes is made. UV radiation is made by use of suitable UV lamp. It is also used in laminar flow clean air bench, where UV tube light is there, and before making any transfer or the laminar flow top, we use the UV tube light for a minute or 5 minutes. Then after some time the environment inside the laminar flow clean air bench becomes sterilized, and therefore we can make the explant inoculation at laminar flow clean air bench. So UV tube light is converting the oxygen available there into ozone, and in ozone no microorganism can remain live. So in UV treatment is also a method for sterilization in laminar flow clean air bench, where we have to make inoculations. The sterilization by filtration, and the filtration technique is a very fine technique, principally used to sterilize solutions of heat liable material, and to avoid all microorganisms present on that particular solutions which are having microorganism, and they are heat labile. The pore size of filters used for filtration is less than 0.75 millimeter, which can be retained small microorganisms, micrometer. This since the diameter of the pores is very small, a suction of or a pressure is essential during filtration. However, it is never possible to be certain that filtration procedures which renders a solution bacterium free will also free it off by viruses also. There are different type of bacteriological filters used in microbiology and in plant tissue culture. The short description of some filters we can have here. The Pasteur Chamberlain candle filter. This type of filter is made by unglazed porcelain. There are different type of filter depending on the pore size. The grids are L1, L1A, 2, 3, 5, 7, 11, 13. The L1 is a coarser and L13 is the finest in the order. These filters are used for removing organisms from fluid to obtain bacterial toxins. The Berkfield filter, this is made by diatomaceous earth, that is algae used for the diatoms which are used for this purpose, and it is pressed in the shape of the candle. On the basis of the porosity, they are variously classified into wheel, normal and dense. Third is sage filter. It consists of a disc of asbestos material through which the fluid is passed through filtration. The disc is inserted into metal holder before use. After use, disc is discarded and fresh one is fitted. The sintered glass filter, this is made up of finely ground glass which is subsequently fused to make the small particles adhere to the each other. These filters is used for sterilization. After sterilization, avoiding temperature extremes. The last filter, that is cellulose membrane filter, generally used for the tissue culture purposes. There are two types of cellulose filters, that is Cerodocol membranes, older type and made up of cellulose nitrate, and the modern type is composed of cellulose. These are the different type of filters used for the enzymes, vitamins and some amino acids, or which are heat labile, some growth hormones like indole acetic acid, or also sterilized by filtration, or some coarse filters. In figure we can see the different type of filters. Here the cellulosic membrane, asbestos filter, the porcelain filters, and treated glass filters. There are different type of filters shown here in the diagram. So these are the different methods which are generally used for the sterilization of different equipment, instruments, explant during the tissue culture and microbiological experiments.